The next speaker is Ignacy Górecki, who is a medicine student, and he will be talking about all the important things related to survival in space, especially the ionizing radiation-induced mitochondrial damage in space. Go on, Ignacy, please.
Thank you, Anya. I'll go ahead and share my screen.
I will let you know 2 minutes before the end of your desired speaking time, so that there is time for discussion.
So let me know if you can see my presentation.
Yep, it's fine.
Okay. Well then, hello, my name is Ignacy Górecki, and today I'll be talking about ionizing radiation-induced mitochondrial imbalance as a potential threat for astronauts. I'm a medical student at the University of Warsaw, and this summer I've also taken part in Young Scientist program at Blue Marble Space Institute of Science, and this is what I've been focusing on during that time. First, a quick reminder of what mitochondria actually are. Well, they are one of the most important organelles in the cell. Most of eukaryotic cells have them, and in human body, only mature red blood cells do not contain mitochondria, and it's because of their function, which is transport of oxygen throughout blood system. And loss of mitochondria in this case is a sort of adaptation to their function. They are called semi-autonomous organelles because, according to endosymbiotic theory of the origin of mitochondria, they were once separate prokaryotic organisms, which then merged with eukaryotic cells, right? So they do have their own DNA, which is functional. It has genes necessary for mitochondrial function, but they wouldn't be able to operate outside of the cell. This is a symbiotic relationship. So that's why they are called semi-autonomous organelles. The main function of mitochondria is to produce energy, to produce ATP in the reaction of oxidative phosphorylation. And that's what we learn at school, but recent years have shown us that mitochondria are so much more than that. They are involved in lipid and sugar metabolism and calcium metabolism. They are the source of reactive oxygen species as a byproduct of ATP production. They are also involved in processes like cell cycle, cell death, or even immune response in some cases. And those are just to name the few. The number of mitochondria in a cell differs depending on what kind of a cell or tissue we're talking about. As I mentioned, some of them do not have any. On average, it's about 30% of cell volume, but tissues and cells with high energetic consumption, like brain, like muscles or liver, are packed with mitochondria. And this is no surprise that mitochondrial dysfunction will affect exactly those organs the most. So people with mitochondrial diseases suffer from neurological disorders such as progressing neurodegeneration, cognitive impairments, epilepsy, or even some psychiatric symptoms. There's also a long list of myopathies that cause muscle weakness, muscle loss, and even disabilities. And there's also a lot of research into mitochondrial dysfunction in cancer development or in aging processes. And I think you can see what I'm getting at. Those are the issues that we see or expect to see in astronauts, especially in the long-time space travel. When you think about it, and look at this image, you'll notice that mitochondrial dysfunction might affect actually other systems as well, like our vision, our hearing, our cardiovascular system. But I think this is even more important for high-performance individuals such as astronauts. Now, let's move to the space environment. And again, a quick overview of what kind of damage is caused by ionizing radiation. First, there is direct DNA damage when those high energetic particles cause lesions in DNA strands, leading to genetic instability and potentially mutations. On the other hand, there's indirect damage caused mainly by reactive oxygen species, which originate from water radiolysis caused by radiation or from mitochondria. And those compounds are highly reactive and can oxidize other molecules like proteins or nucleotides, leading to oxidative stress and inflammation. Now, when it comes to mitochondrial damage and to direct DNA damage, a lot of focus has been put on nuclear DNA, and there's no surprise about it. But nuclear DNA is one place; it's in the nucleus, whereas mitochondria are dispersed throughout the cytoplasm, and the chance of ionizing event is actually higher for organelles such as mitochondria. And mitochondrial DNA is a little different from nuclear DNA. It's circular. And as I said, it does have genes that are necessary for proper function of mitochondria. There are proteins involved in electron transport chain. And there are also things that make mitochondrial DNA especially prone to damage. First of all, mitochondria do not have any efficient repair mechanisms. Second of all, mitochondrial DNA is built in a way that it does not have introns, it does not have histones. So whenever damage happens, some gene will be affected by this damage. And also, mitochondrial DNA is constantly exposed to reactive oxygen species. And what we see in cells exposed to radiation is that this mitochondrial DNA damage leads to decreased efficiency of electron transport chain and increased production of ROS, which further damages the mitochondrial DNA. So there's this vicious cycle of damage there. Because of all of that, the risk of mutation in mitochondrial DNA is even up to 20 times higher than in the nuclear DNA. And knowing all of that, it's quite surprising to see in the real life that in cells that are exposed to radiation, the number of copies of mitochondrial DNA actually increases. We don't know how that happens or why. One theory is that it's a mechanism of compensation for the genes that are damaged, and that might work for some time, but in the long run, it leads to overload of damage in the cell and often to cell death. And now, what happens to those damaged mitochondria? Let's see what our options are. Mitochondria are in continuous cycle of mitochondrial turnover, which are processes designed as a response to stress and changing energetic needs of the cell. So we have biogenesis, so forming of a new mitochondria, fusion, when mitochondria fuse together and form long tubular structures, fission, which is quite the opposite, so fragmentation of mitochondria, when a lot of spherical mitochondria form, and mitophagy, when mitochondria are being removed. So in cells that are exposed to ionizing radiation, what we see at first usually is fission, so fragmentation of mitochondria, which we can see on the image at the 3-hour time point. And this is a moment where sort of a quality control can happen. And things like ATP concentration or ROS concentration or ability of to maintain mitochondrial membrane potential is being evaluated in order to determine the severity of the damage. And if the damage is not critical, those mitochondria start to fuse and create those long chains of interconnected mitochondria, which is beneficial because this provides increased efficiency of electron transport chains, but also it provides an even distribution of energy in the cell. On the other hand, when the damage is beyond repair, well, those mitochondria have to be removed. And what's interesting about that is that mitophagy is actually, or can be a first state of apoptosis. So we see that there is this control of not only mitochondrial function, but also the function of the entire cell system. And this is kind of a neat scenario when everything seems to be in place. But there are other studies that show that actually, after irradiation of cells, new mitochondria are being formed. And this is quite surprising, but let's think about why that might happen. So after exposure to radiation, there's this peculiar state when there's extensive damage, but repair mechanisms are activated and they work like crazy. So the energetic needs of the cell is massive, and that might be a response to that energetic requirements. Also, as I mentioned before, sometimes we see this increase of mitochondrial DNA copies, and this also might be how that happens, because when new mitochondria are being formed, mitochondrial DNA have to replicate. And also there is evidence that proteins that we know that are involved in biogenesis of mitochondria are also taking part in DNA repair mechanisms. There's, of course, a lot of inconsistency in this data. And some say it might be also dependent on the time period of exposure, where acute exposure leads to increased biogenesis, whereas chronic exposure leads to increased removal of mitochondria.
5 minutes to the end of your time. Please leave some time for discussion.
Okay, thank you. But yeah, there's still a lot of mystery and unknown aspects of that. So yes, now the summary and some take-home messages. So mitochondrial dysfunction can lead to serious health issues, which could potentially affect astronauts' performance. Ionizing radiation causes damage not only to nuclear DNA, but also to mitochondrial DNA. And this mitochondrial DNA is actually at higher risk of damage caused by ionizing radiation. Mitochondrial damage triggers activation of mitochondrial turnover pathways. And also, of course, further research needs to be done, as always, especially about the consequences of mitochondrial damage in the long run. So thank you for your attention.
Thank you. Thank you very much for this very interesting talk on the subject that I know nothing about. Well, I know nothing about until your very interesting presentation that told me that there is much more to be worried about when going to space than just like stuff that I worried about before. So yay. If you have any questions about space medicine especially related to mitochondria exposed to ionizing radiation, please let us know. And in the meantime, you discussed it a little bit, but I think that was not totally clear to me. So if you could discuss it a little bit more. Why fusing of mitochondria, which is one of the ways that the cell deals with low levels of damage, what I understood from your description, why does it help to deal with the damage? I'm not sure I understand.
Well, it's also a kind of compensation for the organelles that are damaged, right? So because these electron transport chains are not very efficient in damaged mitochondria, when they fuse, it's kind of a compensating that those electron transport, electron machinery is taking over for the ones that are not very functional.
Okay, but why longer chains or longer mitochondria are better than shorter ones?
This aspect provides the fact that those are chains of long mitochondria provides this even distribution in the cell, even distribution of energy in the cell, because mitochondria do not concentrate in one place in the cell. They are evenly distributed across the cell, so the energy can be provided in distant parts as well as those close to nucleus, which is where usually mitochondria are.
Okay, cool. Thank you. Are there any questions? You don't want to know how to stay alive in space? Come on, that's very useful information. Okay, so I have actually one more question. Like, would those, normal irradiation prevention systems that are, I guess, designed mostly for the prevention of damage of the nucleus, cell nucleus, would they also work, fine for protecting mitochondria, or?
Probably because those, I think you mean the pharmacological countermeasures, or
For
actually,
example, yeah.
Because when it comes to pharmacological countermeasures, now it's easier to find drugs that are those reactive oxygen species scavengers, right? Mm-hmm. So we get rid of those reactive oxygen species. There's less drugs that we have that actually help to increase this activation of DNA damage repair. So yes, those drugs that we have now are actually more helpful for mitochondrial damage than they are for direct damage to DNA.
Okay, thank you. We are trying to get Roberto Parisi to connect to our system so that yes, he's here. Thank you again to Ignacy for his very interesting presentation. And